population is all the members of a single species occupying a certain area at the same time. Microevolution relates to evolutionary change within populations. Population genetics studies the variation in alleles in a gene pool. Moths are diploid organisms and they carry 2 copies of each chromosome. A single gene for body color has 2 alleles, capital D, which is dark color and dominant, and lowercase d, which is light color and recessive. There are 3 possible genotypes for the color gene, capital DD, which is homozygous dominant, capital D, lowercase d, which is heterozygous, and lowercase dd, which is homozygous recessive. The gene pool is the total of all genes in a population. It is described in terms of gene frequencies. The allele frequency is the proportion of each allele in a population's gene pool. The Hardy-Weinberg principle states an equilibrium of allele frequencies in a gene pool using the formula p squared plus 2pq plus q squared remains in effect in each succeeding generation of a sexually reproducing population if 5 conditions are met. The 5 Hardy-Weinberg conditions are no mutation, no gene flow, random mating, no genetic drift, and no selection. The Hardy-Weinberg conditions are rarely ever met, and allele frequencies in the gene pool of a population do change from one generation to the next, resulting in evolution. The Hardy-Weinberg law tells us which factors cause evolution, those that violate the conditions.
In all Hardy-Weinberg equilibrium equations, P equals the frequency of the dominant allele and Q equals the frequency of the recessive allele. P squared is the frequency of the homozygous dominant, 2pq is the frequency of heterozygous, and q squared is the frequency of homozygous recessive. Suppose in a plant population that red flowers are dominant to white flowers. In a population of 500 individuals, 25% show the recessive phenotype. How many individuals would you expect to be homozygous dominant and heterozygous for this trait? Let P equal the frequency of the dominant allele and Q equal the frequency of the recessive allele. Q squared in this equation is the frequency of the homozygous recessive. Q squared is 0.25, so Q is 0.5. P plus Q equals 1, so P equals 0.5. Homozygous dominant individuals are R, or P squared equals 0.25, and this will represent 125 individuals. The heterozygous individuals are calculated from 2pq, which equals 0.5. In a population of 500 individuals, 250 individuals will be heterozygous dominant.
Mutations are permanent genetic changes. Without mutations, there would be no inheritable phenotypic variations among members of the population. Mutations are the primary source of genetic differences among prokaryotes that produce asexually. Genetic drift refers to changes in allele frequencies of a gene pool due to chance rather than selection by the environment. Genetic drift occurs when founders start a new population or after a genetic bottleneck with interbreeding. The bottleneck effect prevents most genotypes from participating in production of the next generation. The bottleneck effect is caused by a severe reduction in population size due to a natural disaster, predation, or habitat reduction. The founder effect is an example of genetic drift where rare alleles or combinations occur in higher frequencies in a population isolated from the general population. When population sizes are so small that there are few, if any, available unrelated mates, interbreeding can occur. Inbreeding does not affect the frequency of alleles and does not cause a population to evolve. Non-random mating occurs when certain genotypes or phenotypes mate with one another. Assortative mating is a type of non-random mating that occurs when individuals tend to mate with those having the same phenotype with respect to a certain characteristic.